Hello everyone. Right now we are going to start the next chapter, and this next chapter is on transcription and translation. And transcription would mean when DNA acts as a template for synthesis of RNA, that is transcription, and when RNA serves as a template for the synthesis of proteins, it is called as translation. So in the last chapter, we covered replication, and in this chapter, we are going to first cover transcription and then translation. Now, both these processes, transcription and translation, they are the major function of DNA. DNA has two functions. Number one, to serve as a template of inheritance that would be passed on from one generation to the next generation, be it mitosis, be it meiosis. And the second is transcription. And this transcription serves as a process, intermediate process for DNA to produce or serve as a template to synthesize protein. So in this chapter, we'll first focus on transcription, and then we'll move to translation. This will be in two parts, transcription and then translation. So two recordings, number one on transcription and then second on translation. And transcription would essentially mean that synthesis of RNA, right? Synthesis of RNA. So DNA serves as a template for the synthesis of RNA. And then RNA would serve as a template for translation. Now, it is not that simple, and I'm sure you all know that there are three kinds of RNAs which work as a team for translation, and these are messenger RNA, then transfer RNA, and then ribosomal RNA. These three RNA molecules, they work in team for protein synthesis. So all the road goes to protein synthesis, translation, right? Now, when I was an undergrad student, I only knew about these three RNAs, but now there are more RNAs, like microRNA, siRNA, guide RNA for CRISPR. So the family of RNA is increasing. So RNA is a really complex molecule, and the birth to RNA is given by the process of transcription. Now, as I mentioned to you in replication, that replication is an enzymatic process carried out by DNA polymerase. Similarly, transcription is also an enzymatic process that is carried out by the enzyme RNA polymerase. Now, they're very different. Although the name sounds very familiar, it's easy to remember, but they're very different. And one of the major differences is that, so in DNA, both the strands, they serve as a template to synthesize a daughter strand, right? Both the template, whether it is lagging or leading, both the strands, they synthesize the daughter DNA molecule. But in case of transcription, only in case of transcription, only one of the strands will serve as a template for RNA synthesis. Only one of the DNA strands will serve as a template, not both. Right? And also, just review what's the major difference between DNA and RNA molecule. So we have covered this. So major differences are: number one, in ribose versus deoxyribose. Second, replacement of thymine by uracil. And another thing is that, just for sake of clarity, so that there is no ambiguity, DNA is a double-stranded molecule, but RNA could be single-stranded, but at certain point it can become double-stranded, right? And then again single-stranded. So this double-stranded RNA here, this is called as a stem, and this bulging structure is called as loop. This bulging structure is called as loop. So RNA could be single-stranded and could be double-stranded. That you will notice in the structures of especially tRNA. So these are the major differences between replication process and transcription, and so on and so forth. But now we will go into more detail and try to appreciate the differences between prokaryotic transcription and translation compared to eukaryotic transcription and translation. So let's go back to the slides. So this is chapter 7, and in the book that is shown here, it is on pages 223 to 258. Right. So let's start. What's the objective of this chapter? What is the importance of transcription or translation? When does transcription and translation, when and where does it occur? And mechanism of transcription and translation? And what proteins, what are the players that support transcription and translation? So these are the objective. And as I have kept the theme that our cells, which is a building block, it needs support from protein, nucleic acid for growth, development, and differentiation. So if transcription doesn't happen, then there are no proteins, and then if there are no proteins, then there is no workforce within our cells that will support such important processes like replication and transcription. Also development. So let's quickly focus on this. So here, if you notice that this is DNA, one of the strands of DNA. I'm sorry, this is one of the strands for RNA. And what is a hallmark of RNA is the presence of uracil, right? Presence of uracil makes DNA and RNA different molecules. Also, on the second carbon atom, if you see, the second carbon has hydroxyl group. This hydroxyl group, the oxygen is missing in deoxyribose. Also, you will see the 4 bases, and thymine has been replaced by uracil, right? So what is the process of transcription? So double-stranded DNA is there. One of the strands will serve as a template for the synthesis of RNA. And then this RNA will support the synthesis of proteins. Okay. Now, within a cell, cell is just like a business house that works on the demand and supply chain. So if there's a protein that is needed in huge amount, like spindle fibers or the cell cycle proteins, or proteins that participates in cell cycle, they are produced in large numbers as compared to some of the proteins that are needed in very low amount at a particular time. For example, tumor suppressor protein P53. So they're not ubiquitously expressed, but they're very selectively expressed in very small amount. Okay. So as I mentioned to you, there are basic three kinds of RNAs. mRNA stands for messenger RNA, that is responsible for the coding of proteins. rRNA is ribosomal RNA. It is a core particle of the ribosome and helps in catalysis of protein synthesis. MicroRNA, and they are small 22 or 23 nucleotide RNAs that regulates gene expression that we'll study later. Then transfer RNA. Transfer RNA helps as a carrier of amino acid for protein synthesis. And where does protein synthesis occurs? It occurs on the surface of ribosome. Right? So, so any translation cannot happen without transcription, and transcription cannot happen without an enzyme called as RNA polymerase. So, just like we have our palm, RNA polymerase is also like a palm shape, and from one end there is DNA that goes on. So RNA polymerase slides across the DNA, and on one hand the ribonucleotides are coming, and from the other end the nascent or RNA is being synthesized, right? But what is the law here? The law is that complementarity, that A will code for U, G for C, T for A, and U for A. Right? Now, this is one of the most important part of this chapter, is to distinguish between prokaryotic and eukaryotic transcription and translation. But we are focusing currently on transcription. And one of the major differences between prokaryotes and eukaryote is compartmentalization due to presence of nucleus in eukaryote, which is absent in prokaryotes. So in prokaryote, there is DNA in the cytoplasm. DNA serves to transcribe, to make our mRNA, tRNA, or rRNA, and then it is translated on ribosomes. So everything happens in cytoplasm in prokaryotes, and genes are very simple. Okay, so DNA will code for RNA transcription and then translation. Now in eukaryote, it is very complex, especially in multicellular organism like human body. So excuse me. So first, the chromosomes, the genes are organized as a mosaic of introns and exons, right? So not all the DNA that is present in the chromosome makes or codes for protein. There could be selectivity. So introns are those regions which don't express protein, but it expresses mRNA or RNA rather. So introns can make transcript, can make RNA, but introns will not translate. Whereas exons will transcribe RNA that could be an mRNA that could serve as a template for translation. So human gene or multicellular gene, they are mosaic of introns and exons. So once again, introns are transcribed, but may not be mRNA. It may not translate, but exons transcribed, they are mRNA and would translate. But when a nascent RNA, when a new RNA transcript is formed, there is a mosaicity of introns and exons. So why are introns there? There should be some function that we will study later. But these introns which do not code for protein, they have to be spliced out. Right? They have to be removed so that exons come together. Now this is not as simple. mRNA, human primary transcript or human RNA, they're very expensive. They're very important. So they have to be protected because in a cell, there could be many ribonucleases. It's very fragile. So it has to gain stability. So the stability to primary transcript, the nascent RNA, the newly formed RNA, comes from two processes. One, capping at the 5' and polyadenylation, addition of polyase at the 3' of the mRNA. So let me explain this a little bit more. So let's say that this is a DNA, double-stranded, and here there is a mosaic of DNA is a mosaic of introns and exons. So let's say the red colors are exons. Right? Red color are exons, right? And the yellow color are introns. The yellow color are introns. Now, when the transcript is made, when the mRNA is made, mRNA will be a mosaic of introns and exons. Now, when this is made, the introns have to be taken out. They're removed by the process called as splicing, and then you have the complete exon. Right. Now before this splicing occurs, there has to be protection of primary transcript. And how the protection occurs? Protection occurs by two processes. On 5' end, there is capping, and on the 3' end, right, there is poly-A tail. So capping and tailing, this helps the primary transcript, the freshly formed RNA, to be stable, right? And then this exon, or let's say mRNA, that comes out after splicing out of introns, then mRNA is exported out to the cytoplasm. Why to the cytoplasm? Because cytoplasm has ribosomes. So due to compartmentalization and the nature of the genes, because of the presence of introns and exons, the multicellular organisms or eukaryote, they have multiple step process in transcription. Whereas prokaryotes, where there is no nucleus, the genes are simple, the transcription is occurring in the cytoplasm, very simple. It's a one-step process. One thing I would like to explain here is that, what I mentioned to you, that DNA is double-stranded. One of the strand will code for mRNA. Right? One of the strands will code for mRNA. So this is the template. This is a double-stranded DNA. This one, 3 to 5 strand, is serving as a template that generates RNA. Now this is very interesting if you notice. So you see the bluish strand and the yellowish strand, and they give rise to an RNA molecule. Now if you notice carefully, the sequence of mRNA or RNA is very identical to one of the strands of DNA. So if this yellow color strand is serving as a template, the newly synthesized RNA is very identical to the strand that is not serving as a template. So that strand is referred as sense strand. And the strand which is coding as a template, that strand is called as antisense strand. Antisense strand. So sense strand and antisense strand. Now let's go into the more deep of transcription. Okay. And what do I say deep? Because we are going into the interplay between the RNA polymerase and the DNA for synthesis of RNA. And this is in bacterial system. Now this is a double-stranded DNA in bacteria. Now this RNA polymerase, to start transcription, has to bind to a unique site, right? Just like on the airport, there's a specific lane or a specific track on which the plane has to land or take off. It cannot do at every part of the runway. So it has to go to a specific runway to take off or to land. So RNA polymerase also land at a specific place on the DNA. That part of DNA is called as promoter, okay? So promoter is the site where RNA polymerase binds, and that promoter is very close, very proximal to the start site of the transcription. So to start transcription, the RNA polymerase has to bind to the promoter. Now also try to recollect what happens in replication. In replication, there's a replication origin, then replication fork, right? Because the whole chromosome or whole DNA has to replicate. But that's not the case with transcription. Not all of the genome transcribed, there is selectivity. So now when RNA polymerase binds to promoter in bacteria, it is not sufficient to start transcription. It needs an additional factor called as sigma factor. Sigma factor helps the RNA polymerase to initiate the transcription, just to initiate the transcription. Once the transcription has initiated, the second stage, the sigma factor launches off. The RNA polymerase do not need the sigma factor. So sigma factor comes out, and then RNA polymerase slides through the DNA to transcribe RNA, right? And only one of the strands will transcribe, right? And then after the RNA is made, right? So unlike DNA, where the replication has to be 100%, transcription is restricted because only that part of the DNA will transcribe, which will code for tRNA, rRNA, mRNA, or miRNA, or any other RNA which has a function. After that, when there's a stop signal, at which transcription has to stop. That stop signal is called as terminator, right? So at the terminator site, the transcription process finishes, and RNA polymerase, everything comes out of the transcription machinery. Right? Now let's go a little bit more in detail. What is a promoter? So what scientists did, they were curious and they wanted to study promoter. So they studied promoters in various species, and they came to a conclusion that there are some sequences in the promoter which is conserved. And one of this sequence is called as theta sequence, T-A-T-A-T-A. The theta sequence, or this is also called as theta box. This is located at -10. This is very conserved amongst various species. So promoter at -10 is very conserved, and the box is called as theta box. The second conserved region is called as -35 region of the promoter. Now, let me go back and explain something to you. So let's say that this is the DNA, right? So this is the DNA, right? Double-stranded DNA. Now, RNA polymerase, right? RNA polymerase cannot start transcription anywhere it likes, right? It has to go to a particular site on the DNA. And that site in the DNA is called as promoter. So this is the promoter sequence. On this, the RNA polymerase will come and bind. The RNA polymerase will come and bind on this promoter sequence. Right? Now, when scientists looked at the promoter, they found some consensus sequence. What were the consensus sequences? One at -10 position, the other at -35. So -10 called as box because it is rich in thymine and adenine. The second portion is -35 region, two region of the promoter. Now the question I'm asking here, or trying to explain to you that what is minus sign over here, right? What is minus site over here? So the minus site is there's a particular site in the DNA, there's a particular base in the DNA which is very proximal to the promoter, which is the starting point for transcription. It is called as start site. Right? Start site. Now, if this is a start site, then this becomes as +1. The start site becomes +1. +1. So any sequence before this will be written as with a minus sign, right? So -10 would mean 10 base pairs ahead of the start site, and -35 would mean 35 bases before the start site. If it is 35 or +35 means after the start site, +35. Okay, so that is the situation with the promoter. And then there's a start site here, +1. And as I mentioned to you, there is a limited size of the protein in terms of number of amino acids. So the RNA has to be of limited size. Transcription is a controlled process. It's not global as compared to replication. So there's a terminator site at which transcription has to stop, right? And this transcription process, termination process, could be of two types. One is row dependent, where row factor comes in to stop the transcription, or there's a row independent process. So depending upon the need for row factor, it could be row dependent or row independent termination of transcription. Now, I mentioned to you there are three kinds of RNA: mRNA, tRNA, rRNA, miRNA, right? So RNA polymerase is a family of proteins. And there are three kinds: RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. And they have a specific role. In what sense? RNA polymerase mostly synthesize ribosomal RNA. RNA polymerase 2 mostly synthesize those RNA that will go and translate into proteins. and miRNA. RNA polymerase 3, transfer RNA plus 5S rRNA. So RNA polymerase, most of the rRNA genes except 5S rRNA. And also small RNAs are synthesized by RNA polymerase 3. Okay. Now, in eukaryotes, it's a complicated process because we have so many cells, multicellular, that cells that differentiate and become specialized, right? So a human body emerges from zygote, and then zygote differentiate. So, and differentiation means from one single cell, you have brain, that is memory, you have intestine, which is digestion, kidney, that is excretory, skin, protective, bones, skeleton. So from one single cell zygote of so many specialized cells emerge. And that is because of orchestration of certain factors during process of transcription and translation. So these are called as general transcription factors, called as TBP, as theta binding protein, TF2D. So these are transcription factors which work with RNA polymerase for transcription. Because multicellular gene, taking human an example, the transcription is a very controlled event. It is not as in bacteria, because in bacteria the cell's objective is just to divide in numbers. But in humans, division is important, but also differentiation. Now I mentioned to you that the primary transcript that is synthesized has to be protected. And there are two processes linked to that. One is called as capping, which is at 5' end, and the other is called as poly A tail. Right? So capping occurs at the 5' of the mRNA, and tailing occurs at the 3'. 150 to 215. Poly A means polyadenine. Many 150 to 250 adenines are added at the 3' of the mRNA. So this is the process of capping. This is the methyl guanosine that is added to the 5'. 5' means 5th carbon atom of the ribose sugar. What is added? 7-methyl guanosine is added to the 5', 5th carbon atom of the ribose. Right? Which enzyme does? It is called as guanylyl transferase. So capping, which is 7-methyl guanosine, addition is an enzymatic process that occurs at the 5' with the help of enzyme called as guanylyl transferase. Similarly, addition of polyadenine nucleotides occurs at the 3', and this is carried out by an enzyme called as poly A polymerase that adds catalytically poly A tail to the 3' end of the mRNA. Now, it is as compared to, as I mentioned to you, bacterial gene, which is mostly coding region that synthesizes or transcribes RNA. The human has a mosaic of introns and exons, and introns, they transcribe, but they don't translate. So for efficiency, introns have to be removed. Introns have a very interesting role in regulation that we'll study later. But here, let's focus on the process of splicing. So the blue colors are exons, right? And the orangish or yellow color is intron. So I want you to focus on this adenine residue, right? This adenine residue in the middle of introns play a very important role in splicing. And why does it play? So what is there is that this adenine has this adenine nucleotide, the second hydroxyl group of this nucleotide. It serves as it begins a nucleophilic attack at the junction of this intron and exon. So this is the junction, right? The yellow is intron and the blue is exon. So at the junction, this OH group from adenine makes a nucleophilic attack. Because of this nucleophilic attack, this OH is exposed, and this spliced intron goes and self-ligate to the adenine, right? So this sort of becomes the starting point for lariat shape. And then this hydroxyl group again attacks at the junction of exon and intron, right? And then after this OH group and phosphate from this, they form a gene, and this intron is spliced up. So I have briefly written here that you can read for more understanding. After this, the mRNA is ready to shuttle into the cytosol. In the cytosol, it tries to go to the ribosome for the translation. Right? And John O'Keefe, British American scientist, will share half of the for getting awarded for in medicine for discovering transcription and process like that. So again, coming back to the same story, this is most important. So transcription, then addition of capping and tailing, and then splicing, and then export. Now in some bacteria, there are a special sequence called a Shine-Dalgarno sequence. Shine-Dalgarno sequence that helps the bacteria, the mRNA, to go and attach through hydrogen bond to the ribosomal RNA, 16S ribosomal RNA. So Shine-Dalgarno is a specific signature sequence for bacteria that helps mRNA and rRNA to interact with each other on the surface of ribosome. And this will be the beginning, but here I would end this chapter, transcription, and then the next recording will be on translation. Okay.